Well, it's really nice to be here to talk to you all this evening, and thank you very much for the invitation. It's a great privilege. So I'm going to tell you a little bit about my botanic garden, the Cambridge University Botanic Garden. And in particular, I'm going to introduce it to you generally and then tell you about its place in the story of evolution and its relationship to Charles Darwin, which seems appropriate for this evening's talk. I'm then going to talk a little bit more about botanic garden living collections in general and how they work and how they're used to support research, and particularly research into evolution. And then I'm going to tell you a little bit about some of my own research at the end, which is about an understanding, in this case, of iridescent flowers, so how flowers can look different colours from different angles, and how that work can be driven by the living collection of the botanic garden. So I'll start by introducing the Cambridge University Botanic Garden. I'm sure that some of you are familiar with it, but I imagine most of you aren't. So here's my sort of standard go-to slide on my botanic garden. And people are often surprised by the first bullet point there, which is that it's a research and teaching collection of 8,000 living plant species. So people sort of expect you to start by saying it's a park or it's 40 acres, but actually the important thing is that it's a collection of living plants which are used for research and teaching. And that collection, as I'm going to show you, is very much more like a museum collection, as is every botanic garden collection, than like a park or a public, or something like an RHS garden. So the collection is at the heart of the botanic garden. That collection supports a global network of researchers. So we provide our 8,000 living plant species to researchers from anywhere in the world who want to access that material for their work. So we send them leaves or seed or DNA, whatever it is they need to support their research. Now, my living collection is arranged in a 40-acre landscape, and it's a heritage listed landscape. It's a very beautiful landscape. And it would be a shame to collect 8,000 plant species in 40 acres and not use them to show to people and let people enjoy them. So we support an education programme for all ages, from preschool to adult, and we also run interpretation trails and various events which showcase the garden, its work, and the importance of plants, plant science and horticulture in the world generally. And visitors are, of course, a very important part of any botanic garden, and we have 345,000 visitors in 2022. So it's a well-visited site. Now, the key thing about a botanic garden, and the thing that separates it from any other type of garden you might visit, are the labels. And that is why I really want to explain these to you at the beginning. So the labels are the things that link the plants on the ground to the database we hold. And that's what makes it a collection, like a museum. So just as every item in a museum isn't just there randomly, but is recorded in a database and has a label and an accession number, so do my plants in the botanic garden. So here's a standard plant label, and this is the accession number I'm showing you in the top left. Accession number is a code. It actually tells me this plant came into the collection in 2017. But the key thing is the number, and that links it to the database where that number will tell you about this plant. We show its Linnaean name, its common name, where it came from, what family it's in, and the little code at bottom right tells me where the plant came from immediately prior to coming to the botanic garden. So W means it was wild collected, this plant was collected in the field. But the important thing is the succession number, because when those researchers around the world write into the botanic garden asking, do you have Potentilla erecta for my research? And rather than having to walk around 40 acres wondering whether we've got any and looking for it, we simply go to our database, we check, it said yes, it tells us the key, the accession number, it tells us where in the collection it's kept, and we're able to go and get it and send it on to the researchers. So in principle, just like every item in the museum collection should have a label and a code like this that links it to a database, so should every plant in a botanic garden. And my curator likes to say that every label should have a plant and every plant should have a label. There should never be a separation of the two. Now, if you're a museum curator, that's a little bit easier than if you're a botanic garden curator, because your museum collection doesn't move in the night. It doesn't die. It doesn't set seed, which grows and germinates somewhere where you didn't put it. And so actually a large part of the work of running a botanic garden is about maintaining that link between label and plant and making sure that our collection and our database and our labelling in the flower beds is actually up to date. And that's quite a challenge. But it's the key thing that makes a botanic garden special. Now, that's a very brief introduction to the Cambridge University Botanic Garden. I'm going to say more about it over the course of the talk, but I'm going to tell you a little bit now about its place in the story of evolution. I think it's important to get that link to Darwin in early on. So Cambridge originally had a different botanic garden than the one that we have today, and this is one of the few paintings we have of the original botanic garden. If anybody knows Cambridge, this is actually King's College Chapel here in the centre of the picture, and that's right in the centre of town, and that's not where the botanic garden is today. So this original botanic garden was founded in 1762, and like all botanic gardens of the time, it was a physic garden. So that's a garden which was used to teach medical students in the university how to identify and use plants to treat human disease. So there'd be a flower bed of plants that were used to treat conditions of the heart, and a flower bed of plants that were used to treat conditions of the liver, and so on. And medical students would be taught to identify them, how to extract the key compounds, how to use them. And botanic gardens began as physic gardens. This was the origin of the botanic garden idea across Europe. So this is ours in 1762. The earliest botanic garden was that in Padua, founded in 1545, again on those same sorts of principles. And if you know the Oxford Botanic Garden, which obviously I don't like to mention, but it is there, it is still very much that physic garden design. So it's much older than the Cambridge University Botanic Garden, and it has, so 400 years old, and it has that very traditional physic garden design. Now, we don't have that in Cambridge because we had a professor of botany in the early part of the 19th century who understood and realized that plants were far more important than just medicine for people, and that they had far more potential in biological research. So this is a man called John Stevens Henslow, shown here. And Henslow was the professor of botany in the university from 1825. And he persuaded the university that actually plants had a lot to teach us about how life worked generally, that they were fundamentally important biological organisms, and he couldn't study them in a tiny couple of acre plot full of plants that were thought of as medicine. He wanted lots of space, he wanted to be able to grow a much bigger collection of species, he wanted to be able to grow trees, which are not traditionally grown in physic gardens, and he wanted land to do that on. And so he persuaded the university to buy him our 40-acre plot, which at the time was outside Cambridge proper. It was fields owned by one of the colleges, Trinity Hall, and rented to a local farmer. Of course, now, as Cambridge has grown, we're very much in the centre of town, we're just next to the train station. And we have the plan here on the left from 1846 of the garden, and it's very recognisable to any visitor coming to visit today. So we still have the lake, which is this sort of serpentine shape here. We have our systematic beds here, which I'll talk about later. We have a woodland area here. And we have this lovely curving path around the outside, with the trees, as you see on the map, planted in family groups around the outside, because Henslow was interested in understanding the relationships between different types of plants. Now, when I say that Henslow understood that plants were more important than just as medicine, he really was ahead of his time, because one of the things he argued to the university to persuade them to buy the site was that many of the fundamental discoveries of biology would be made in plants. And I want to show you briefly, just with a few examples, that he was absolutely right. So, for example, before Henslow's time, the fact that organisms are made up of cells was actually discovered in the 17th century by Robert Hooke, looking at his wine bottle cork down a microscope. Of course, what he was looking at in his wine bottle cork was the bark of the cork oak tree, and he discovered that it was made up of cells. But following Henslow's time, Gregor Mendel's famous work in genetics, the discovery that traits were inherited in a particulate way rather than in a blending fashion, was done with peas. He actually started to work with chickens, but peas were easier to work with. Perhaps less familiar to people is the idea that stem cells, which we hear about in the news as medical discoveries, were actually discovered in the 1950s in tobacco. And perhaps more esoteric ideas, more familiar to biologists, are jumping genes, which are common in all plant and animal genomes and create mutation, were discovered in the 1980s in maize, and the more recent discovery of gene silencing, the idea that genes can be switched on and off, was discovered in the 1990s in petunia and tomato. So Henslow was very much ahead of his time in understanding that plants were going to sometimes be just the best organisms to study the basics of life in, and that if you wanted to do that, you needed to have access to a good supply of them. Now, his own research interest was in the nature of a species and in the meaning of variation. So Henslow was working and studying before The Origin of Species was written, before Darwin, and he was interested in trying to understand if a species had been created to have a particular form, how far could you stretch that form? How much variation could you tolerate before you were no longer that species? And we can see that thinking all around the Botanic Garden in his planting. So here, for instance, is a tree. is a tree, a pair of trees, a pair of beeches that we still have on site, planted in 1846, which are grown side by side. And the one I'm showing you on the left here, as you look at it, has a cut leaf form, and the one on the right has a round leaf form. And this is Henslow planting them side by side and saying, well, how come they look different if they're the same species? Similarly, here are a couple of black pines from our main walk. The one on the left is a snow shedding form from the Austrian Alps, and the one on the right is from Corsica, and sort of sundays or sprawls across the path. It's the same species, and Henslow's planted them side by side as he tries to understand, how does a black pine come to look different in these two different locations? We also see this in his herbarium collection. So Henslow was a great botanizer. He would go out into the Cambridge countryside at the weekends and collect plants to make herbarium sheets. And usually when you make a herbarium sheet, you put a single pressed dried individual on it. But you see here, this is Henslow's Timothy grass sheet. He's got multiple individuals, from a tiny one here to a very tall one here, a very bushy one here. He's collected the range of variation within the species within a single population, and he's pressed them together as he tries to understand, well, how is a Timothy grass supposed to look, and how far can we stretch that? Now, this is really interesting research, I think, but it's not what he's most famous for. He's most famous as the person who taught Charles Darwin when Darwin came to Cambridge to study theology. So Darwin, in fact, attended all of Henslow's lectures, which was supposed to be very interesting, and they went out together at the weekends botanizing, and Henslow taught Darwin how to identify plants, how to study them, and how to press them and make those herbarium sheets as well. And it's impossible not to imagine when you're looking at the trees in the Botanic Garden in Cambridge and thinking about these ideas of variation and how far you can stretch the limits of a species. It's impossible not to think about the conversations that the two of them were having during those walks, during those study sessions, and how that thinking and those discussions led to the thinking that Darwin did, of course, later on. And it was Henslow, as Professor of Botany at the University of Cambridge, who was asked by the government to nominate a naturalist to go on the voyage of the Beagle. And he writes to Darwin, I've been asked by Peacock to recommend him a naturalist as companion to Captain Fitzroy. Of course, he nominated his student, Darwin. One of the things I particularly like about this letter, though, is how it goes on. Let me show you the next bit, where he says, I state this not on the supposition of your being a Finnish naturalist. Very important, even if your student's going to turn out to be one of the most important and famous scientists of all time, to keep them in their place in those early days and make sure that they know that you still think they've got work to do before they're a Finnish naturalist. So Darwin went off on that voyage at Henslow's suggestion. He thought what he thought and he wrote what he wrote. And he also collected an enormous number of herbarium specimens while he was out there. And he sent all of those back to Henslow, to his teacher and mentor, Henslow. And we have all those still in the Cambridge University Botanic Garden in the herbarium. So the University Herbarium is based in the Botanic Garden. And we have these wonderful specimens of the Sikios velosa here, which was collected by Darwin, is extinct in the wild and is only known from this single herbarium sheet. And there are other wonderful sheets, this one where, at Henslow's request, Darwin's folded the leaf frond over so that you can see both sides of it. So I do hope and encourage you to visit Cambridge sometime to come and see the Botanic Garden, and also to get in touch and we can arrange to show people the wonderful herbarium as well, where all of those specimens are preserved. So that was a little bit then about the Botanic Garden in Cambridge's place in the story of evolution and our link to Charles Darwin. I'm going to say then more about how Botanic Garden living collections are used today. Because of course, as I said at the beginning, it's a living collection that's the heart of what a Botanic Garden is. So this is a view from a drone looking down on that systematic beds, just a small part of our living collection, but you get a feel for how a Botanic Garden is organised and the wealth of species that are within it. Now I'm going to tell you a little bit about our collection, and I want to sort of think about numbers for a minute to try and give you some context for it. So Cambridge University Botanic Garden has 8,000 species, 14,000 accessions, so that means that we have multiple varieties or subspecies or types of some of those species. We have 9 national plant collections, so that means we're the national collection holder for 9 different collections, and that makes us the largest national collection holder in the UK. And we have the most diverse arboretum in the east of England, about 2,000 trees. There are more diverse ones, of course, on your side of the country, but in the east we have the most diverse collection. Now, 8,000 species might sound like a lot, but I want to encourage you to try and think about it within a context. And so actually, the first question you might ask is, well, how many plant species are there? What percentage of the world's diversity of plants are you collecting? And the answer to that is a little bit disappointing. So actually we have 8,016. And that comes up out at slightly under 2% of all of the plant species on Earth. There are over 400,000 plant species. So that doesn't look very impressive looked at like that. But then those 400,000 plant species are spread all across the Earth. So then you might ask yourself, well, how many do you normally find in a given area? You might also ask yourself about how they're distributed among families and orders and other taxonomic groups. So let's just look at that one first and tell you that our 8,000 species cover 44 of the 47 orders of plants, 243 of the 463 families, and about 2,000 genera of about 14,000. So when we're thinking about our collection, we can say, well, actually, it'd be quite nice to fill this gap and collect some of the orders that we're missing, maybe even some of the families. We're unlikely to fill all of the genera. What about how common those species are in botanic gardens generally? Well, that's when things get really interesting, because our 8,000 species actually represent an enormous biodiversity hotspot. The UK flora is less than 3,000 species. So we've got nearly 3 times the total flora of the UK in our 40 acres. If you look at countries that you might think of as biodiversity hotspots, well, Congo has about 6,000 species, we have 8,000. Madagascar has about 9 or 10,000 species, we have 8,000. So actually, it's a lot of plants in a very small area. But to make those most valuable to researchers and scientists, we want to make sure that they're the plants that they want to work with. And we also want to make sure that they're plants that they might not easily be able to obtain anywhere else. So if we want our collection to be valuable to scientists, there's not a lot of value in growing something that you can buy in every garden centre in every town in the UK. Scientists could just go and get it down the road. What we want to be able to offer them are species that are harder to get access to. And so one of the pieces of work that my curator, Sam Brockington, has been doing for the last few years has been cross-referencing our collection with the global network of Botanic Garden collections. And we can do that through an umbrella organisation called Botanic Garden Conservation International, which allows us to access the databases of other botanic gardens. And through that, we've taken our 8,000 species on this graph here. So here they are on the bottom, species 1 through to 8,016. And we've asked how many other botanic gardens grow them. So you can see that species 1 is grown by more than 200 other botanic gardens in the world. Now, that's probably not going to be that useful to researchers, or at least it may be useful, but it's not uniquely useful. They can get it in lots of other places. Whereas down at the other end of the graph, there are 357 accessions that no other botanic garden has. So if a researcher wants to work with them, they can only get them from us. And so one of the things we've been working on, focusing on, is thinking about how we can extend the tail of the graph here to provide researchers and scientists with access to plants that they can't easily get in other places. Now, to do that, we need to think about the sort of research that our botanic garden might support and what sort of species are likely to be most useful. We're never going to be able to grow all 400,000 plant species in 40 acres. So to show you some examples of the sorts of research that people do with a botanic garden collection, well, one thing is that they focus on biodiversity loss. And so this is a project called the DarwIN Tree of Life project. It's funded by the Wellcome Trust, and it's mostly running out of the Sanger Centre in Cambridge, where the human genome is sequenced. And what the DarwIN Tree of Life project aims to do is to sequence the entire genome of every plant, animal, fungus, and eukaryotic microorganism in the UK. So that's an awful lot of organisms. And of course, what we can do as a botanic garden is make sure that they have access to the right plant material for that. We can supply properly taxonomically validated plant material for that sort of project, which is aiming to understand the biodiversity of the country as a whole. We can also work with plants that aren't from our own country. So this is Amorphophallus titanum, the Titanarum, in flower at Cambridge Botanic Garden about 6 or 7 years ago. These plants flower very infrequently in the wild. So they flower once in their life and then die at about 15 years old usually. And they are critically endangered in their native Sumatra. It's one of the largest, it is the largest unbranched inflorescence in the plant kingdom. And the way it attracts pollinators is to produce an appalling smell of rotting flesh from the chimney in the middle, which heats up to about 37 degrees centigrade to make those smells go up like a chimney into the rainforest and attract carrion beetles and carrion flies. Now, it's wonderful to be able to show this plant to people, but the other sort of research we can do is actually to improve our understanding of it and to improve, to improve our understanding of it and to improve our ability to preserve it and protect it. So I'm just trying to change my okay. Never mind, the slide was going to show you it heating up to 37 degrees centigrade, but we'll just have to trust me for now. Oh no, it will work. So if you watch it and you see the people are pale coloured, the plant in due course at the top heats up to the same temperature as the people were. And so the top of that column gets to 37 degrees centigrade to release volatiles. And using this thermal imaging camera, we were able to understand the time frame that happened in, and also which parts of the plant were responsible for the production of the heat, which is really important for understanding how it works in the wild. But the other thing we were able to do was pollinate it. So it requires cross-pollination from another individual. We were able to get pollen from colleagues at the Edinburgh Botanic Garden and pollinate our Titanum, produce all of these little fruits. And we now have about 150 of them sitting in our glass houses. And we are working to spread those to other botanic gardens around the world so that other botanic gardens can share the excitement and the educational opportunities with people. But we're also working with colleagues in Sumatra to reintroduce them into the wild. It's really important with a plant like this that's endangered that we're able to do that. We have the ability to propagate it in this way. We also, of course, are often supporting research that's involved with climate change, so it's intrinsically linked to biodiversity loss. But for instance, this alpine, the saxifrage, lives in a range which is likely to be lost. So as climate warms, alpine plants move up the mountain and the ones at the top have nowhere to go. So studying its range and the temperature ranges it can tolerate is an important part of being able to understand that. And also understanding the minerals it produces from its leaves, which are an important part of its survival strategy. So supporting this kind of research is very important to us as well. And it's trying to predict this kind of research that we need to do if we want to make our collection as useful as possible. Our collection is also used not just for research, but also for education. It's easy for me to get focused on the research and forget to tell you about this really important part of what a botanic garden does. But for instance, we have adult trails which take on board scientific thinking. So this one talks about plants and climate change and takes people on a tour of the botanic garden looking at plants which are likely to respond in different ways to climate change, and plants that could also be used to mitigate climate change in different situations. So this is a really important part of the use of the collection too. The third sort of research that we support, that I wanted to mention in this context, is into food security. And a really important part of the work of many botanic gardens at the moment is in providing access to researchers to plants that are relatives, wild relatives of different crop species, so that scientists can try and understand how they grow, how they might tolerate different soils to the crops, how they might resist different diseases than crops do. If we can understand that in the wild relatives of crops, then it might be possible through breeding programs and other approaches to actually improve the ability of our crops to withstand different climates, different diseases and so on. And so again, having access to that living collection of different species is really important to allow researchers to do that. Now, bringing all of this thinking together has led us, and primarily this is the work of my curator, Sam Brockington, to develop a living collection strategy for Cambridge University Botanic Garden, set for 2020 to 2030, unfortunately published just before the COVID pandemic struck, which has delayed our ability to implement it for a couple of years. But the idea behind this collection strategy is that we have done a lot of the thinking into what sorts of plants researchers are likely to want to access, what sorts of plants we're most able to grow and most able to provide value through providing access to, and then thinking about how we bring them into the collection over the next 10 years to make our living collection as useful as it possibly can be for the sort of scientific research that I think with Henslow. Now, it's quite challenging to do this. There are 400,000 plant species. We can only grow 10,000 maybe of them. We want to choose as well as we can. And one of the difficulties of choosing, I've just talked to you as if it's something that can be rationally designed by thinking about research into biodiversity loss and climate change and food security and so on, is that actually the diversity of things that plant scientists work on is enormous, and the questions they ask can't always be predicted. And so I want to actually tell you about why I study plants and what I find interesting about them, and that will lead me into talking to you a little bit about my own research as well. So my reason for studying plants, and it's a reason that quite a lot of my colleagues I think would share, is encapsulated in this picture. And if I ask you what the picture is of, I'm sure that most of you would say it's a picture of an elephant. But actually, from my point of view, it's not a picture of an elephant at all. It's a picture of maybe 30 different species, several hundred different living organisms. There's a broadleaf species here. There are quite a few individuals of some pink flowered species here. There are at least 3 or 4 individuals of this white flowered species here. There's quite a lot of individuals of at least one grass species down here. There are multiple different tree types going on the trunk colours here, and so on. Pink flowered shrub at the back here. And there's an elephant. So there are lots of different species, lots and lots of different individuals. People tend to see the one elephant. But actually, if you think about the lives that these different individuals live, the elephant has it really easy. So every individual in this picture has the same set of problems. They all have to get enough to eat. They have to stay warm enough and dry enough, but not get too dry, not run out of water, and not get too hot. They have to find a mate. They have to set their offspring off into the world with enough resources to get started on their own. They have to avoid being eaten. And the elephant's solution to all of these problems is just to walk away. It can walk to somewhere drier or cooler, wetter or warmer. It can run away from a tiger. It can walk towards a potential mate. The plants, every single one of the plants in the picture has to solve exactly the same set of problems, but it has to do it rooted to the spot. And that means the solutions that the plants come up with to these problems have to be so much cleverer. They have to come up with a whole range of different solutions to the same problems, but without the movement aspect. And it's understanding those solutions, in my case, the solution to the problem of finding a mate, so pollination, that makes plants so fascinating to me, but also to so many other researchers as well. And because of the enormous diversity of strategies that plants have to come up with to these problems, it can be quite hard, quite challenging to predict what species researchers are going to want to have access to. So that leads me then to telling you a little bit about my own research into understanding floral iridescence. And I'm going to start by telling you, setting the scene and explaining the background to this work, and then a little bit about some of the science we've been doing lately. So the background to this work is in the context of pollinators and agriculture. So you'll be familiar, I'm sure many of you will be familiar with this quotation at the top here, which you see in the news a lot, that every third mouthful of food we eat relies on pollination. Now that statistic, it's not really a statistic. It comes from a paper, or a set of papers, which have shown that 35% of global food production by volume comes from crops that depend to some extent on animal pollination. And the extent to which those crops depend on animal pollination varies. So if you look at something like beans, actually you get about 90% of the yield anyway, even if there are no pollinators. Pollinators add about 10% to the yield of most beans. If you look at something like kiwi fruit, on the other hand, it's the opposite. You get almost no yield at all, no kiwi fruit without pollinators. And then if you look at things like apples or raspberries, it's very variable according to variety and also where you're growing the plant. So the yield loss can be as high as 90% as low as 40%. And the map I'm showing you here shows you how that actually plays out across the world. So you can see by the colour coding what percentage of the agricultural GDP of each country is dependent on animal pollination. So for us sitting here in the UK, it's about 5%. But for some countries, it's much higher than that. So you see it's very large indeed, 20% for China. And there are some small pockets where it's higher still. There's some actually chocolate production here at over 20%. Whatever the place you are sitting in this data set, though, it's clear that animal pollination is really important for food production, and understanding how animal pollinators produce fruit crops and seed crops and oil crops and nut crops is really important in being able to feed a growing population. And there are many different interactions that flowers have with animal pollinators. So I'm showing you here a variety of flower crops that have very different relationships with their animal pollinators. So something like the beans or the oilseed crops here are actually only dependent 5 to 10% on pollination, on animal pollination, but they're grown to such an enormous extent across the world that 5 to 10% equates to an enormous amount of calories, of protein in the case of the beans, and indeed of money. So there's a lot of value in that 5 to 10% of the yield that's related to animal pollinators. Then there are other systems. Here's chocolate, which I showed you. It's very important, really 100% dependent on animal pollination. Here's vanilla, which is entirely hand-pollinated at the moment as a commercial crop, and that's extremely expensive in terms of labour. Those of you who've grown cucurbits and melons or cucumbers in your garden will know that the flowers are actually what we call monoecious. They're either male or female. So they're 100% dependent on animal pollination. They can't possibly self-pollinate. And then things like Brazil nut here as well. Very much like chocolate, very, very strongly dependent on animal pollination. But I hope you can see by looking at them that these flowers all differ in fundamental traits, shape, colour, size, pattern, and that all affects how different animals interact with them. So my research group is focused on understanding how key traits of flowers affect animal pollination. And I want to understand how those traits are built so that we can think about whether we can breed them or engineer them into crops. And I want to understand how they work. So I'm thinking about things like colour, size, symmetry, texture, whether there's a tube or a spur, and how that causes the relationship with different pollinators like the bumblebee or the honeybee here to differ. Now I could talk about any of these traits today, but I've focused on one, and I'm going to talk to you about a trait that you may not have thought about, which is iridescence of flowers. So I'm going to start by introducing my favourite iridescent flower to you. This is Hibiscus trionum. And what I want you to see is that there are 5 petals. They're predominantly white, and at the base they have a patch of purple pigment. But I hope you can see on your screen that the lower 2 petals, that patch appears blue, and it doesn't appear blue on the upper 3. Now that's not because the petals are different, they're all the same. It's because the colour, this blue colour, is angle dependent. You can only see it from certain angles. If I tilted the flower away from you on the screen, the blue colour would shift up to the upper petals, and you would see those as blue instead. So when a colour is angle dependent, we call it iridescent, and you're most used to seeing angle dependent or iridescent colour in something like the soap bubble. And the soap bubble lets me point out to you that actually you can't make iridescent colour using a chemical pigment like the one that's making my jumper, this orangey brown colour. You can only make it from a transparent material that interferes with light. In the case of the soap bubble itself, you know, is transparent. It's the thin film of the soap bubble interfering with light that produces the colour. Now, people often think they haven't seen or noticed many iridescent flowers, so let me just show you a few. Here's a tulip. This is the Queen of the Night tulip. The dark brown purpley colour is pigment, but you can see, I hope, this iridescent sheen on top of it. So there's yellow and green and blue colours, and that's caused by a similar effect to what you've just seen in the soap bubble. Here's a daisy that's doing the same sort of thing as my hibiscus. So we've got angle dependent blue here at the base of the ray florets. You can see it on some and not on others. But actually, very often when we're looking at iridescent flowers, we find that it's hard to see unless we take the pigment out of the equation. So this is actually the skin of a yellow flower that to my eye just looks yellow. But if I peel the skin off and put it on a microscope slide and then change the angle with which you're looking at it, you can see it's reflecting different colours of light at different angles, so red, yellow, blue, as I move it around. Now this ability to make colour from a transparent material and colour that changes with angle is something that we're more familiar with in animals. So here are a range of animal species that are using the same sort of effect, and we call it structural colour, and it's made by a structure rather than pigment. So the rosemary beetles that you might see in your garden, their red and green stripes are made through a transparent material interfering with light. The blue morpho butterfly is perhaps the most famous example. The blue here again is a structural colour, not a pigment. But you also see it on bird feathers, fish, spiders, and so on. Now, the structure that's making this colour on the flower is called the diffraction grating. And you're probably most familiar with the diffraction grating from looking at a CD or a DVD. So when you look at a CD, you can see the rainbow effect, the iridescent colour. And that's because a CD has data groups. So I'm showing you them here on a scanning electron microscope. And they're spaced at about 1 micron apart. And that's about the same size as a wavelength of light. The result of this is that when white light arrives at this structure, it's broken into different colours and reflected at different angles. So you get this rainbow effect produced. Now, if you look at an iridescent tulip on the same magnification, you can see that we have the same system of ridges and grooves, spaced again at about a micron apart, and they're able to produce colour. That colour isn't as dramatic as from a CD, and that's because if you look in cross-section at the tulip, I hope you can see that's a wide gap and that's a narrow gap. There's a little bit of irregularity or disorder in the system, which you wouldn't get in a man-made CD, and that makes the colour a bit less good. I'm going to come back to that later in the talk, because less good to my eye and your eye doesn't necessarily mean less good to a bee's eye. So these diffraction gratings, or ridges, or striations, are made up of a material called cuticle. And the cuticle is the outermost layer of a plant cell. So here are two plant cells side by side. They have this cell wall. You might remember this from biology at school, but there's a cellulose cell wall around plant cells. And if they're epidermal cells on the surface of the plant, they'll also have this cuticle, which is a hydrophobic matrix of chemicals that basically stops the water in the plant cell being sucked out on a sunny day. It's a waterproof coating. But the waterproof coating on my iridescent petals has this ridge structure, and we've been trying to understand how we build that. Now, to understand that, we looked at how the flowers develop. And so we broke our hibiscus flower development down into 5 stages. There are the petals at those 5 stages. And we looked at the cells in the white region and the purple region of those 5 stages. And we discovered that the ridges form between stages 3 and 4. So between stage 3 and 4 here. And they form at the same time as the cells change shape. So up until stage 3, the cells are what we call growing isotropically, so equally in all directions. But at stage 4, they shift to anisotropic growth. So they're growing more in the y-axis of the screen than in the x-axis. And we wondered if there was a relationship between the shift in cell growth and the formation of wrinkles. And to explore that, we started talking to colleagues in the engineering world. And they told us that absolutely, there was a hypothesis in engineering that could explain this. And that was the hypothesis of mechanical buckling. So starting with the diagram on the left, they said if you take 2 materials that are bound together at an interface and you shrink them, there's nowhere for this upper material to go, so it will buckle and form a system of ridges and grooves like the one on your head. And we said, well, that's very interesting, but nothing's shrinking when a flower grows. Things are growing, not shrinking. And they said, well, there's a way of explaining that as well. I'm hoping I can run this video. I was able to run the last one. It doesn't want to. Yeah. So if you watch what's happening in this video in 1 minute, we're dropping a drop of ethanol, a drop of alcohol, on top of a bilayer of material. And the ethanol is absorbed by the upper layer of material, but they're bound together. So there's nowhere for this suddenly expanded material to grow. And so instead it buckles, and all of these tiny little lines here are buckles or ridges. Now there's no water to them, there's no stretching of them as there wouldn't be in my growing petal. And as the alcohol evaporates, the material goes back to smooth. And so the idea is that if you actually have a 2-layer material and the top layer grows faster than the lower layer, then you get the same effect as the shrinkage, and that could give you the buckling. And so we've developed a mechanical model which says that if you take into account the elongation of the cells, the deformation of the cell as it grows upwards, the stiffness of the cuticle and the cell wall, and the growth rate and thickness of the cuticle, then in theory you can generate these buckled ridges and the diffraction grating without needing any other components to the system. So it's a nice simple model. It takes into account things that all plant cells have. It doesn't require any new newly evolved genes or any new systems. But of course what we want to do next is test our model. And we would test the model by modifying the amount of cuticle that the petals produce, so how fast that upper layer grows, modifying the growth of the cells, so the stretching, and modifying the stiffness and chemistry of the cuticle, because those are important in order to allow the buckling to occur. And we've been working on testing that model over the last few years. And we do it using a technique called genetic modification. Now, you're used to hearing about genetic modification as a potential agricultural tool. Plant scientists use it to actually explore how genes work and how plants grow. So we've been modifying hibiscus by infecting it with a soil bacterium called agrobacterium, which transfers genes into the plants, and then we can grow little plants up that contain genes that we put in, and those can be genes that do things like change the amount of cuticle. So here's the normal hibiscus. Here's one where we've added a gene that reduces the cuticle, and we've lost the ridges. We can also change the stiffness of the cuticle. So here's our normal hibiscus. Here's one where we've played around with the stiffness of the cuticle, and here's one where we've made it much, much less stiff, and by changing an enzyme involved in cuticle production, and we've lost the ridges completely. And I hope you can see that this flower has lost the blue effect, so we've lost the iridescence. So these techniques are helping us to understand how you build iridescence, but I'm also interested in understanding why it's there. And to do that, we use a separate lab setup, which is our bee behaviour lab. So in this setup, we buy boxes of bumblebees, colonies of Bombus terrestris, the buck-tailed bumblebee, which are sold for pollination of tomatoes and strawberries in polytunnels. We connect them with a gateable tube to what we call a flight arena. And we let the worker bees come out through the tube and forage in the flight arena. And when they come to us, they've never met a flower. And so we can train them on anything we like. This is a standard artificial training flower. So it's a test tube with a pink lid. And there's a little cup on the top with 20 to 30% sugar solution in. And the bees forage for the sugar, take it back through the tube to the box and feed it to the queen and the larvae in there. And while the workers are out foraging, we can paint mark them. So I hope you can see there's a little blue spot on this bee, and a little red spot on this bee. And that means we can follow individual bees. So I can say to one of my students, it would be really good to know what the first 100 choices that 20 independent bees make is. And they can go and collect that data. Because if you study bees in the wild, you're never sure whether you're looking at the same bee you saw 5 minutes ago or a different bee. Of course, if it rains, you don't get any data at all, whereas this setup works in all others. So I'm going to show you data from just 2 experiments to explore how iridescence affects bee behaviour. So in the first one, we've set up an equilateral triangle of artificial flowers, just plastic discs, in our flight arena. They've all got a cup of sugar on. And if you imagine that we've got 3 of these discs and they're all blue, what we do is we time how long it takes a bee from leaving the first disc to landing on the second, and leaving the second disc to landing on the third. And if I do that enough times for one bee, I can tell you how quickly that bee finds blue flowers. And if I do it for lots of bees, I can tell you how quickly bees find blue flowers. And then if I add a diffraction grating to the surface of that blue plastic disc, I'll print one on top and repeat the whole experiment, I can tell you how quickly bees find blue iridescent flowers, and therefore what effect the iridescence has. Does it make it easier to find the flower or harder? So let me show you the data from one of these experiments. This is actually the red artificial flowers, and the bars 1 and 2 are the first flight and the second flight. So with non-iridescent flowers, it takes you 4 or 5 seconds in this setup to find a flower. If we add a diffraction grating, it takes you about 3 seconds. So iridescence speeds up foraging. That's good for the bee and good for the flower. But the problem here is that this experiment was done by printing the surface of a CD onto the red discs. And as I showed you a while ago, a CD is perfectly regular and a real flower isn't. So we were a little bit concerned real flower iridescence isn't as impressive to my eye. Is it going to be as effective to bees? We repeated the experiment with a replica of a tulip printed on the surface of our discs and found actually floral iridescence is just as good, or even better, depending how you do the statistics, at improving foraging efficiency as perfect iridescence is. So floral iridescence is perfectly good enough for the bee's eye. It leads to a significant increase in the ability to find a flower, and that's good for the bee and good for the flower. But we wondered whether it would have any negative consequences, because one of the ways bees work is that they form search images of flowers when they're out foraging. So if you watch a bee on a patch of lavender, it will go from lavender flower to lavender flower to lavender flower. It won't fly after a buttercup. So they form a search image of a good rewarding flower. And you might think that iridescence, because it changes colour when you change the way you look at it, would make that harder to do. So here's a different experiment. What we've done is train this bee through several hundred visits that blue flowers are always good and always have sugar in them. And then we introduce some slightly differently coloured flowers, so some purple flowers. Now, if she's formed a good search image of blue flowers, she shouldn't go to the purple flowers, she should ignore them. But if she's not been able to form a good search image, she'll go to both. And again, we do this experiment without a diffraction grating and with a diffraction grating and see what the difference is. So Let me show you again the effect of an artificial perfect diffraction grating. So the data is set up to show correct choices out of 20. So in this situation, the bee can correctly identify the rewarding flower 70% of the time, 14 out of 20, if it doesn't have iridescence. But when we add a CD replica onto the surface, that drops down to only just over 50%. It's almost having no ability to form a search image at all. So the iridescence really does damage search image formation. But that's with a perfect CD replica. If we do it with a flower replica, there's no effect at all on search image formation, but he still gets it right more than 70% of the time. So unlike perfect iridescence, that slightly less good floral iridescence doesn't compromise search image formation. So if we put those two ideas together, floral iridescence is as good as perfect iridescence at increasing salience and therefore foraging efficiency. It makes flowers stand out and easy to find. And it's as good as no iridescence at making sure floral signals are clear and easy to remember. And in combination, that makes it perfect to the bee eye, even if it isn't as impressive to my eye or your eye as a CD would be. It also means it's not surprising that iridescence has evolved multiple times across the flowering plants. So if you look at a phylogenetic tree, a family tree of the flowering plants or angiosperms, every one of these orders I've marked in red is an order in which we find these diffraction gratings independently evolving. And that sort of convergent evolution perhaps isn't surprising, because I've showed you that you make these ridges out of things that all plants have, and that they have such a positive effect on pollinators. But I think it's the sort of discovery that Darwin would be quite excited about. And from my point of view, it's the sort of discovery that can only be done if you have access to a botanic garden with a really great living collection, so that you can actually study all these species in. And so, of course, what we're doing now is going back to our wonderful living collection and exploring how those different individual species make the ridges, is it through the same model, and how they affect pollinator behaviour. It's wonderful to have access to the living collection to do that work. Now, I'm very much running out of time, so I'm going to finish up by saying thank you to the members of my research group that have actually been involved in this project. And I'm lucky enough to work with a great team of postdocs and PhD students. And it's these three here, Jordan Ferrier, Chiara Aroldi, and Carlos Lugo, who've been particularly responsible for the iridescence work I've shown you. We also work with a range of great collaborators and have support from a number of wonderful funding bodies as well. So I must thank all of those. I'll finish with one of my favourite views of Cambridge University Botanic Garden. This is the Indian clock vine in our tropical house. Encourage you to come and visit it if you ever get the opportunity. And thank you very much indeed for listening to me this evening.